So today I am going to discuss with you all about the alternative splicing. Well, we have done about the splicing process in early videos. Now let us discuss about a unique pattern that our cell follows to make different types of proteins, different isoforms of proteins using the same mRNA, and that process is called your alternative splicing. So let's get started now. So what it says about alternative splicing here, that alternative splicing, it is a process by which exons or portion of the exons or non-coding regions within the pre-mRNA transcript are differentially joined, or sometimes these exons or non-coding regions are skipped, that results in multiple protein isoforms, means different types of the proteins will be formed that will be encoded by a single gene. So there are multiple exons, there are multiple introns present within them that has to be spliced out, that we have discussed during splicing process. But here what happened, in alternative splicing we make a combination of different exons. Okay, some exons retained in that mRNA during the splicing process. Sometimes some non-coding regions remain within the transcript to make a multiple types of isoforms of protein from that single gene. That is your the concept comes of alternative splicing. Okay. Now this alternative splicing mechanism increases the informational diversity, different types of proteins, diversity of proteins being produced from that same gene, and functional capacity of a gene during post transcriptional processing, and provides an opportunity for gene regulation. So this splicing process, this with the help of this alternative splicing process, we increase the different types of proteins from encoded by that same gene. We make different isoforms of protein, and the functional capacity also during post transcriptional modification processing, and provides an opportunity for gene regulation also. It also have a concept of gene regulation. By this technique we can also able to regulate a particular gene also. Okay, next it is saying that alternative splicing generates a tremendous amount of proteomic diversity. The same thing has been written that we have said. Proteomic diversity means different variants of that proteins can be created in humans and other animals, and significantly affects various functions in the cellular processes, like in specific tissues, in developmental states, and disease conditions. So, alternative splicing generates a tremendous amount of proteomic diversity. It creates different variants of the protein that significantly affects various functions of the cell types, tissue types. Okay, just like in case of for making proteomic diversity, we are different ways like alternative splicing, RNA editing. Okay, uh, different ways are there to make diversity in humans, so to make diversity of proteins in living organisms, so that they function differently in specific tissues, specific cells, or depending on developmental states and disease conditions. Okay, various examples are there. We will discuss one by one in the videos. Okay, let us move to the next page. So, this is the concept of your alternative splicing. So, you can see here a genomic DNA, and during transcription we form a pre-mRNA. You can see a pre-mRNA here, 5 prime, 3 prime, multiple exons are there, and between the exons there are introns are also present. So by the splicing process, we can remove of these all introns and join the exons by true two trans-stratification reaction that is mediated by the spliceosome complex. Sometimes they mediated by itself if they are of category, if the introns are of group 1 introns or group 2 introns that we have studied before. But we are studying here about alternative splicing. So what happens during alternative splicing? You can see here, these are the exons. You can see 1 2 4 5. Okay, again I repeat, 1 2 4 5. So the exon number 3 has been skipped here. Exon number 3 has been skipped here to make a unique different RNA transcript. From that different protein product will be produced. Okay, so mature mRNA where the exon 3 has been skipped here. So it is a isoform of your mRNA, okay, of this particular mRNA to make a protein isoform, which is a functional mRNA. Okay, function A refers to, suppose this protein where the exon 3 has been skipped will have a unique function in specific tissues or in specific cells or different function in the cell. Then you can see on the right side your, here also the exon has been skipped. Here exon 4 has been skipped. 1 2 3 5, no exon 4 is there, and thus we form a new type of mature mRNA which will be further translated to form a protein of different types from the same mRNA, function B. And also we also have a normal protein product where all the exons will be remaining in the mRNA, all the introns will be removed. Sometimes introns can be retained within the mRNA, like for example intron 4 remain, it is not spliced out, whereas all the other exons has joined to form a unique mRNA where exon number 1 2 3 4, even the intron sequence is also present, the introns is present. Okay, then 5. Okay, okay, wait for a minute. Okay, so these are 5 prime, these are 3 prime. So we make a unique protein now. We make a unique protein means isoform of the same protein. Isoform of the same protein we can create. So various ways can be done. Well, here you can see the dot 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 that I have shown. It shows that exon 2 is joining with exon 4. Okay, mutually exclusive exons, and exon 3 has is more compatible to bind with exon 5. So there are different types of alternative splicing. We will discuss maybe on the next video. Okay. So these are general concept of alternative splicing. Okay. Now we will little bit say about the types of alternative splicing also. Okay. So I hope you have understood this part. This is the basic thing of alternative splicing, how alternative splicing occurs. Okay. Moving to the next page. Okay. Now I have given an example. Various examples has to be mentioned here in order to explain, in order to identify the alternative splicing. Well, you will also find out here many things like the enhancers, silencers, you will also find out. So an example of alternative splicing, your mammalian muscle protein called troponin T. Okay, so this mammalian muscle protein troponin T, the mRNA, the pre-mRNA from the troponin T has 5 exons. That means before the formation of this troponin T, which is the muscle protein, mammalian muscle protein, the mRNA which encodes this troponin T, better to say the pre-mRNA, which has not been processed now, it has 5 exons. Okay, very good. And this mRNA, that encodes the troponin T is spliced out to form two alternative mature mRNA. That means the mRNA that is encoded for that troponin T muscle protein, it can be spliced in two ways, and it has five exons, okay, but it is spliced in two alternative ways to form two different types of mature mRNA, and each mature mRNA will have four exons, just like I have said, I have shown to you in the previous diagram. It will have four exons present. A different exon is eliminated from each of the two mRNA so that the two messages have three exons in common, as well as each carrying one unique exon. So here we can see a simplified diagram of the pre-mRNA of um, not pre-mRNA, sorry for that. The middle one is the pre-mRNA, the primary RNA transcript for the troponin alpha troponin T, alpha troponin T. This one is your pre-mRNA. Okay. Now from this pre-mRNA, you can see the exon 1, 2, 3, 4, 5, and the orange ones which shows here, right here, these are the introns. Okay, good one. These are the introns. Some small, some large. Now you can see here, I have just said that this troponin T has five exons. Okay, we can see here five exons, and the mRNA is spliced here in alternative ways. Here the mRNA is spliced in alternative ways to create two different mature mRNA. So we can see here two different mature mRNA is being produced. But each of these mature mRNA has four exons, not five exons. So one exon has been missing. Better to say not missing. One exon has been forced to skipped out. Okay, so this mRNA will have two alternative mature mRNA is spliced out to form two mature mRNA, alternative mature mRNA having four exons. A different exon is eliminated. You can see here the exon number 4 has been eliminated. Exon number 4 has been skipped, or you can say eliminated in this picture, whereas in this bottom figure exon number 3 has been eliminated, exon 3 has been skipped. Okay, so from this you can see a unique pattern of mature mRNA is being produced. Okay, as you can see here, we have named him. The first figure that we can see where the exon 4 has been skipped, it makes a protein called alpha troponin T. That means the alpha subunit of the troponin T, which is a muscle protein found in humans, in mammals. Whereas the exon 3 has been skipped to produce such type of mRNA which will give rise to the beta form of troponin T. So in this way, from one gene, from one single mRNA, by alternative splicing, we are making different variants of the protein, isoforms of the protein. Okay. Other examples are also there, like one example is there. For example, here it is not written. Here it is not has been written. SLO gene. Okay, SLO gene. Now this SLO gene has many exons. It is spliced in many alternative ways to give hundreds and hundreds of exons. Okay, another example is the DS scam gene. Now this DS scam gene found in the Drosophila, in the fruit fly, in the fruit fly, that is Drosophila, whereas SLO gene found in the rat. DS scam gene is also can form from this gene. Variants of proteins can be prepared by the process of alternative splicing. I have shown only the muscle protein of troponin T found in mammals, where it is spliced in two alternative ways. But DSCAM gene is alternative spliced in millions and millions of ways. Sorry, not millions, thousands of ways, to create different isoforms of that same gene to make a unique protein. Okay. Now let us move to the next page. Now it is written here that, I hope you can able to see it. It's a little bit haziness. Alternative splicing can arise by various number of means. Okay. Alternative exon being chosen. Exon can be extended or deliberately skipped. Also, introns can be retained in some mRNA rather than being deleted again, generating a diversity in the protein produced. So, alternative splicing can arise by various reasons. Not reasons, here it is saying that by various ways alternative splicing can be carried out. The first process, it is saying, exons can be extended. That means there is a exon, it can be extended. It can be extended means it can be extended with along with the intron, because just after end of each exon there will be a intron sequence, then again there will be a exon sequence. So exons can be extended. It simply means that we are continue to have this intron sequence with exon, some of them. So exons can be extended or deliberately be skipped. Skipped means, eliminated. That exon can be eliminated. So two ways can be here it is saying. Exons can be extended or it can be skipped. It can be missed out. Also, introns can be retained. The whole introns can be included. That means it is not spliced out. It is not spliced out. It is included or in some mRNA rather than being deleted, and generating a diversity in the protein produced. Okay. So here is the picture which has been shown. Here a simplified diagram has been shown where I have shown just 3 exons and 2 introns to show the process of alternative splicing, the types of alternative splicing, where the orange ones here a little bit yellow is become it, but it's the orange color. These are the introns, where the black portions are the exons. Well, this is the DNA form. When it is transcribed to form mRNA, which is a single strand, you can see the exons written by E1, E2, E3, and the introns written by I2 and I1. Now, what are the alternative ways of splicing? Let us see. First, it is saying normal. Okay, that means the exon 1, exon 2, exon 3 are fully present. This is the normal way of splicing that we have seen in our previous classes. The second way it is saying is exon skipping. Well, exon skipping means some unique exons, particular exon is skipped, as we have seen in human mammalian muscle protein troponin T. Okay, troponin T has been spliced out in two different ways. To make alpha troponin T, the mRNA that we get from the alpha troponin T, that mRNA, its exon 4 has been skipped out. Whereas to for the production of beta troponin T, exon 3 has been skipped out. Here we can see exon 2 has been skipped out. Only 1 and 3 is present. Okay, so this is a second way of splicing that we call as alternative splicing. Third way is exon retained. Well, exon retained means to continue with the exon. That means just after one exon number 1 there was intron sequence, but some of the intron sequence are remaining with the exon 1, this portion. After exon 1, intron 1 sequence has been retained within the mRNA, mature mRNA. That is called exon retained. Exon, what it says? Exon retained. Yes, exon retained. Means to continue with the exons, the introns are continued along with the exons, but to some extent. Next is your intron retained. Intron retained means to remain. Okay, that means the intron is fully remaining within the mRNA transcript. Here we can see no splicing of the intron 1 has been done, but the other introns has been removed. And another is alternative exons. Alternative exons means, just we have seen, combination of exons are remaining together, mutually exclusive exons, you can say 1 and 2, 1 and 3, or 2 and 3. Okay, so these are the different ways of your alternative splicing. We will see one by one in little bit in detail. Okay, now there are some things which has to be mentioned here. Okay, uh, let us discuss in detail. So let us see. It is saying here that some splice sites are used only some of the time, leading to the production of different versions of the mRNA from different transcripts of the same gene. So it is saying here clearly that not all the exons, not all the splice sites, the 5' splice site or the 3' splice site that we have seen in splicing process, that is found within the intron exon boundary, these splice sites are used, some of the splice sites are used in specific time, like during development or in different tissues, that lead to the production of different versions of RNA from different transcripts of the same gene. Okay, so that's why the splice sites are not used every time for the splicing process. That's why we have seen that some introns has been retained here. That means their 5' splice site and 3' splice sites are non-functional at this time. Okay, now alternative splicing can be either constitutive or regulated. It can be either constitutive splicing or regulated splicing. Regulated by whom? By the operators. Well, it can be regulated by your operators. Operators. It can be regulated by the promoters. Well, by the promoters and by the enhancers and silencers. By enhancers or by silencers. So alternative splicing can be either constitutive or it can be regulated. Regulated by the help of operators, which acts as an on-off switch, as we have seen in the operon. By the promoters, we all know that within the promoters, it provides the platform for the mRNA, for the RNA polymerase and the other transcription factors to bind and continue the transcription process. Or alternative splicing can be regulated by enhancer and silencers. Enhancers, as we have studied before, that enhancer, it allows, it actually, what can I say, induces the transcription of that specific gene, processing of the specific gene. Or silencers, well, here the enhancer or silencers are related to the splicing process. It will allow the spliceosome complex to come and bind or not to these splice sites, right? So just we have seen in the previous below the ASR proteins, right? the SR proteins, which is serine arginine rich proteins, they bind to such sequence called what are the name of the sequence we have seen before. Uh, it is a type of enhancer, exon splicing enhancer, ESE. So SR proteins bind to the exon splicing enhancer regions, and these SR proteins actually allows the binding of the spliceosome complex to identify the 5' splice site and the 3' splice site and binds. So it is a type of enhancer. Okay. So one example I have given, SR proteins now. We will see them, okay, in this lecture. So just like that, there are also regulators also, which are silencers, which will silence the region. Let us see. So it has been saying that alternative splicing can be either constitutive or regulated. In constitutive splicing, more than one RNA product is always made from the transcribed gene. So in constitutive alternative splicing, more than one RNA product will be formed, will be made from the transcribed gene, as we have seen in human troponin T muscle protein. In case of regulated splicing, different forms of RNA are generated at different times under different conditions or in different cells or in different tissue types. So, in constitutive splicing, more than one RNA product will be always made from the transcribed gene. Just like that, exon skipping, exon retaining, intron retaining, uh, okay, in that way. But in case of regulated splicing, different forms of RNA will be generated at different times under different conditions or in different cells or tissue types. That means it is fully controlled by the promoter, by the operator, by the enhancer, by the silencer, by the activator and repressors. Enhancer silencers are the regions, right? Enhancer and silencers are the region where the activators and repressor will bind and regulate the whole splicing process here. One example I have given, the SR proteins, the serine arginine rich proteins, which is acting as a activator. It binds to the enhancer regions and allows the spliceosome complex to continue the splicing in that particular site. So first we will discuss with the help of example about the constitutive splicing. Example of constitutive alternative splicing is seen with the T antigen of a monkey virus SV40. So one example of a constitutive alternative splicing that is normally seen with the T antigen of a monkey virus SV40. Let us see what it's saying about this virus. It is saying that, wait for a minute. Okay, it is saying that the T antigen gene, which is found in the monkey virus SV40, the T antigen gene encodes two protein products. One is called large T antigen, that is capital TAG, and the small T antigen, which is small t Ag. So the T antigen of the virus called SV40, it has two protein products, two isoforms of the protein, large T antigen gene and small t antigen gene. And these two different isoforms of proteins are occurring as a result of alternative splicing, just like troponin T I have said. So these two proteins occurs as a result of alternative splicing from the pre-mRNA of that same gene. Okay, so this large T antigen and the small T antigen protein product is produced as a result of alternative splicing from that same pre-mRNA of that gene. Okay, so here we can see a few things between the intron and exon. Well, we have seen about the 5' splice site and 3' splice site before, but some more things we have to see here. Well, how this 5' splice site found here? It's because of some sequence which is unique to the 5' splice site. There is also 3' splice site here also. Okay, to clarify this doubt, we will see here. So it is saying that the T antigen has two exons. Okay, it has two exons. In the mRNA encoding large T antigen, exon 1 is skipped. Sorry, exon 1 is spliced out directly to the exon 2, deleting the whole intron sequence that lies between them. So again I repeat, what happens in large T antigen protein? As I have said that T antigen has two exons having the 5' splice site and 3' splice. This is a 5' splice site. In case of large T and small T antigen, it will be used later on. So I will not I will not now it say this one. So it has 5' splice site and 3' splice site. There is also a stop codon here also, which will terminate the translation process here. There is an intron sequence between these two exons. Okay, now it is saying, let us see. T antigen has two exons. Okay, the mRNA which will encode large T antigen, the mRNA which will encode the large T antigen, in that mRNA, exon 1 will be spliced directly to exon 2, and it will delete the whole intron sequence that lies between them. Usually that we see in normal splicing process that exon 1 and 2 will be joining and removing that whole intron in the form of lariat ring. This is seen in case of production of large T antigen. Okay. Now the mRNA encoding the small T antigen, the mRNA encoding the small T antigen uses a alternative 5' splice site. That is the 5' splice site which is just located near to the 3' splice site. If you see the picture carefully, as I have mistakenly I have said before, you can see a 5' splice site. An alternative 5' splice site is also present close to the 3' splice site, right? This 5' splice site will be used to make small T antigen gene, to make small T antigen protein from the mRNA. Okay, so again I repeat, for the large T antigen protein, the mRNA that is used for the large T antigen protein, they splice these whole introns and join the exon 1 and 2, right? But in case of preparation of small T antigen, they use this 5' splice site. This is an alternative 5' splice site. Already there is a 5' splice site close to the intron exon boundary, but there is an alternative 5' splice site which will be used for making small T antigen gene. Okay, small T antigen gene. But you can say mRNA, better to say. So it is saying here that the mRNA that encodes small T antigen, they uses a alternative 5' splice site, that is the 5' SST, which is located just close to the 3' splice site. Thus in this case, the mRNA includes some of the introns as well. So in this case, the mRNA includes some of the introns as well. That means simply means to make a small t antigen gene, they will use what can I say? They will use this as a 5' splice site, and they will use as a 3' splice site. So this intron will remain. This intron will be remaining in the. So this is the exon. Suppose exon 1. Okay, wait for a minute. So this is your exon 1, this is your exon 2, and some of the introns will be remaining, as we have found of alternative 5' splice site, to make a small t antigen gene, to small t antigen mRNA. From this small t antigen protein product will be produced. So the mRNA that uses small t antigen will use a alternative 5' splice site, which is located just close to the 3' splice site. Thus in this case the mRNA includes some of the introns as well. So this is a clear an example of an extended exon, where you can see the exon intron boundary is not being used. An alternative 5' splice site is used instead of using the normal 5' splice site, and some of the introns remain during the splicing process after splicing to make a unique uh, matured mRNA that will code for the small t antigen gene. So the larger mRNA encodes small protein because the introns is not present. The large mRNA will encode smaller proteins because there is an in-frame stop codon within the region of the intron retained in the mRNA. So obviously you can see here that there is a stop codon also present within the intron regions, right? So this whole as the in so I am writing now for you to clarify. For the large T antigen, we can see the whole introns has been removed out. Okay, but for the small T antigen gene, this is used as an alternative 5' splice site. So alternative 5' splice site, which is located close to the 3' splice site, is used for splicing process, and thus some of the what can I say? Okay, some of the intron is remaining. Some of the introns remain within the pre-mRNA. Okay, so this is your exon 1, this is your exon 2. But don't think that from this large mRNA we will get a large protein. But why we say a small t antigen? This will give rise to a large t antigen gene, but this will give rise to a small t antigen gene. Why? Why this will give rise to a small t antigen gene? It's because if you look closely to this intron sequence, there is a stop codon here. I am zooming in. There is a stop codon here which is located within this intron region. So during the process of translation, when the ribosome will identify these as stop codon, immediately your translation process will be stopped and a small polypeptide chain will be produced that give rise to small t antigen. So in two alternative ways, your this t antigen of the SV40 virus is being produced. In one of the case, we see the two exons are joined to make a large T antigen gene. In another way, we see that this is used as a 5' splice site. So this one and this one is joined to make a mRNA which will code for small t antigen. Now, why small t antigen is being produced is because due to the presence of this stop codon, it leads to the formation of truncated or immature protein product, which leads to the formation of small t antigen from this spliced mRNA. So this is a clear example of extended exon. Okay. This larger mRNA encodes smaller proteins because there is a stop codon between the intron regions that is retained in the mRNA. Okay. So I hope you have understood what I have said this part about this constitutive splicing with the help of an example, that the larger T antigen remain where the exons is joined, where there's a small T antigen gene is being produced from the mRNA by using alternative splicing. They use this 5' alternative 5' splice site, which is located close to the 3' splice site. As a result, we get a larger mRNA. But as within the intron which is retained as a result of alternative splicing, within this intron there is a stop codon also, and due to which your small mRNA is being a small protein polypeptide chain is being produced, that we call as small T antigen gene. Now it is saying that both forms of the T antigen are made in the cell infected with SV40. So when a cell is infected with the virus SV40, both forms of this T antigen gene will be observed. The large T antigen gene and the large T antigen protein and a small T antigen protein. But the ratio of the two forms produced does not differ depending on the level of splicing protein SF2 ASF. Okay, so there is a splicing protein called SF2 ASF. Um, it differs. The ratio of the two forms, that means the large T antigen protein that is formed from the mRNA and the small T antigen protein that is formed from the mRNA, it has to be spliced out before it has to be spliced out, then only you can translation will be produced. So it is spliced out by using the SF2 ASF protein, which is a serine arginine rich protein. So SF2 ASF recognizes these 5' splice site and 3' splice site within the virus SB40 virus genome, and they use the splicing process to make these different forms of mRNA. So SF2 ASF is a serine arginine rich protein. When this SF2 ASF present at high level, then this protein directs the machinery to favor the use of closest 5' splice site and thus produce more of the small T antigen mRNA. So it simply means if the level of SF2 ASF is more, then what will happen? If the concentration of SF2 ASF is more, then they will try to use the closest 5' splice site, and the closest 5' splice site is the 5' splice site which is located just close to the 3' splice site, and thus most of the case, if the level of this SF2 ASF is serine arginine rich protein is high, then more and more small T antigen mRNA will be produced. Well, these SR proteins, as I have said before in the last video lecture, um, so it is saying in this case here that the concentration of the small T antigen mRNA will be produced more if the level of these serine arginine rich proteins known as SF2 ASF is more, because they will try to use the closest 5' splice site, which is an alternative 5' splice site located close to the 3' splice site. Here I have said about the SR proteins, the serine arginine rich proteins. So the here in this case, serine arginine rich proteins bound to the specific sequences. Obviously, the serine arginine rich proteins cannot bind here and there. It has to recognize certain sites, certain enhancer regions. So the serine arginine rich proteins bind to the specific sequences around the exons known as EAC. EAC means exonic splicing enhancers. So within this exonic splicing enhancers, these serine arginine rich proteins bind. After binding, these SR proteins will interact with the splicing machinery and thereby recruiting them to the nearby splice sites. This is the reason that why at high levels of SF2 ASF protein directs the machinery to use closest 5' splice sites and also produce more of the small t antigen mRNA. So what happened here is, if I look at the diagram, you can able to recognize. So I have said that if the level of SR proteins, if the level of serine arginine rich proteins, here the serine arginine rich protein is SF2 ASF, right? Here the serine arginine rich proteins in this case of monkey SV40 virus is your SF SF2 ASF. This SR protein bind to certain regions called exonic splicing enhancers. And when the concentration of this type of proteins are high, what they will do, they will bind to this exonic splicing enhancers nearby. So it binds here. This SR proteins binds here, SR proteins binds here close to the site, the alternative 5' splice site. It does not bind here. Where they find more, they bind more. So they will also bind here also. That's not a problem. But close to the sites they will bind if the concentration become more. And thus by binding to the exonic splicing enhancer, they will allow or recruit the spliceosome machinery to come, the spliceosome complex to come and bind and grab on and carry out the splicing process. That's why this is the main reason that why your high levels of this type of SR SF2 ASF protein, that is your serine arginine rich protein, leads to the uh, splicing of the alternative 5' splice site and leads to the production of more mRNA, which will codes for small t antigen gene. Okay. Okay, so now some regulators, some enhancers, some silencers which has to be done, has to be said now. So what it is saying, proteins that regulate the splicing process binds to the specific sites on the exons and on the introns. There are some proteins which bind to the specific sites during splicing process. Okay, and it regulates the splicing process. And those specific sites where the proteins bind, like the SR proteins bind, I have just now said, those specific sites are exonic splicing enhancer or ESE or intronic splicing enhancer or ISE. If the proteins interact with these enhancer regions, either in the exons or in the introns, then it will enhances the splicing machinery nearby the splice sites. Another site is their silencer, exonic splicing silencer or intronic splicing silencer. If within this region the proteins bind, SR protein or other regulatory proteins, if they bind, then it will repress the splicing machinery to come and bind to continue the splicing. It will prevent the splicing process to do. So this is the case that where how introns are retained sometimes, exons are retained sometimes. Okay, so there are some sequences, there are specific sites where these regulatory proteins bind. One is called enhancer regions. Enhancers, here the exonic splicing enhancers, which are found within the exons, and intronic splicing enhancer, which are found within the introns. Binding to these sites, binding to these enhancer regions will allow the splicing machinery to continue. There are some also other sequences called exonic splicing silencer or intronic splicing silencer. Binding of the proteins to this silencer region will repress the splicing process. Okay. Now these sequence elements are proteins are very important in directing the splicing machinery to many exons, even when alternative splicing is not involved. Okay. One of the such enhancer is SR proteins, serine arginine rich proteins. Serine arginine rich proteins have different names, like I have said, the SF2 ASF, which is found within the monkey virus SG40. It is an example of SR rich proteins. The SF2 ASF is an example of SR rich proteins. So SR rich proteins are large and diverse. It has specific roles in regulating the alternative splicing by directing the splicing machinery to different splice sites under different conditions. So these serine arginine rich proteins are large, they are diverse. It has different roles in regulating the alternative splicing by directing the splicing machinery to different splice sites under different conditions. Okay. Now, if I generally speak the structure of SR rich proteins, we will see that it has two important domains, it has two important regions. One domain where it will interact with the mRNA at the enhancer region, at the silencer region, and another domain which will interact with the proteins of the splicing machinery. So, just what I have said, SR proteins has two domains. In one domain, it has well characterized RNA recognition motif. That means with the help of this domain, with the help of this important functional region of SR protein, they will recognize the specific RNA sequence. It will bind to the specific RNA sites. Other domain of the SR protein is the RS domain, which is a serine arginine rich domain. This RS domain is found at the C terminal of this SR protein. With the help of this RS domain, SR proteins can interact with the splicing machinery. Thus, it can recruit the splicing machinery to that splicing sites. So, SR protein, if I say all of the SR proteins is a type of a regulatory protein. It is a type of an activator. You can say an activator always bind to the enhancer regions to enhance the splicing process here, where the enhancers we know the exonic splicing enhancers ESC and the intronic splicing enhancers ISC. If the SR protein bind to this enhancer region, it will allow the splicing machinery to come and interact with this and binds to the 5' splice site and 3' splice site or alternative 5' splice site found within the intron to carry out the splicing process. Okay, so we have seen here that SR protein has two domain. It has two important functional region. One is called RNA recognition motif, where the SR protein will interact and bind either to the enhancer or to the silencer. Other domain is used to interact the SR protein with the splicing machinery. Okay, so with the help of this RS domain, SR proteins can interact with the splicing machinery, where it can recruit the splicing machinery to a nearby splice site. Okay, so SR protein is an example of an activator, and I have discussed about the activator in the previous classes. In the offline classes, I have discussed. So normally activators will allow the induce the process here. It will induce the splicing process here. Okay. So SR protein is an example of an activator if MCQ question comes. Okay, and all of these activators like SR proteins have an RS domain, whether it is an SR protein, whether it is an other activator protein, they have a RS domain which will helps to interact with the splicing machinery proteins to recruit within the splice sites, and SR protein will interact with the enhancer regions, EAC or IAC. There is also some repressor proteins as well. Repressor protein like ISS, ESS. Sorry, not repressor proteins ISS. ISS and ESS are the sites, are the silencer sites. Intronic splicing silencer, exonic splicing silencer. Okay, Now, now within this silencer region, the repressor protein will bind and will repress the spliceosome complex to interact and bind at that sites. So repressor do not have this RS domain. If you see the repressor proteins, there are different examples of repressor. I will do one of the example. Repressor do not have this RS domain. That means the RS domain, which is a serine arginine rich domain, it does not have. Thus, it does not able to recruit the splicing machinery to that site, and they block the specific splicing sites, and it repress those sites. So simple thing, in this way this the splicing process is regulated by the activators and by the repressor. Here the activators I have said, an example I have given, the serine arginine rich proteins or SR proteins. An example of serine arginine rich protein is the SF2 ASF, right? So this serine arginine rich proteins, which is an example of an enhancer, which is an example of an activator, they bind to the enhancer regions like exonic splicing enhancer, intronic splicing enhancer, and when they bind, they recruit the splicing machinery to come to interact with the splice sites at that region. Whereas oppositely, conversely, repressor are the opposite of activators. Repressors, they also bind, but they bind to the silencer region like intronic splicing silencer, exonic splicing silencer, and after binding, they, as they don't have that serine arginine rich domain, repressor cannot able to recruit the splicing machinery to come and interact at that splice sites. Thus it, and it also blocks the splice sites by binding. So thus repressor, in this way it blocks the splicing process at that particular region. In this way the splicing process is regulated by the help of these two different types of protein which binds to these splice sites. Okay, one more example has been given about the activator. Well, I have discussed about the activator here, like the SR proteins. Let us try to discuss about a repressor. Okay, let us try to discuss about a repressor now. How the repressors uh, prevent the splicing process. Okay, most silencer called intronic splicing silencer, exonic splicing silencer are recognized by a member of heterogeneous nuclear ribonucleoprotein family. So in short, the heterogeneous nuclear ribonucleoprotein family is also called as HNRNP. They are a type of what? These are a type of repressors. These are an example of a repressor, the HNRNP. So these HNRNP proteins are actually repressors. They bind to the RNA. Specifically, they bind to the silencer region. They bind to the silencer region like intronic splicing silencer, exonic splicing silencer. And because these repressors don't have a serine arginine rich domain, as these repressors don't have this serine arginine rich domain, they lack this domain, so they cannot able to recruit the splicing machinery to come and bind to the silencer region. Not silencer region, they prevent the splicing machinery to interact with the 5' and 3' splice sites. They prevent, and instead they what they do, they block that specific splice sites and repress the use of those sites. So the same thing is done by the repressors to bind to the silencer, but instead it does not recruit the splicing machinery to come and interact with the splice site because they block the splice sites. So thus the splicing process is prevented. Okay, so simple thing, exonic splicing enhancer, intronic splicing enhancer are recognized by the activators like the serine arginine rich proteins are all activators. They come and bind and allow the spliceosome complex to come and interact with on that region where the 5' and 3' splice sites are present and carry out the splicing process. But here those which are repressor, most of the repressor are heterogeneous nuclear ribonucleoprotein family, which is HNRNP family. They are repressor. They actually bind to the silencer region, and after binding to the silencer region, they actually block the splice sites so that the splicing machinery cannot able to come and bind. Thus, they repress the splicing process. And why they don't allow the splicing machinery to come and bind? Because they don't have this RS domain, they don't have this serine arginine rich functional domain, so they cannot able to recruit the splicing machinery. Let us see an example of a repressor. Okay, then we can able to clarify our doubt. I think after hearing this whole lecture you can able to understand about this uh, regulation of the splicing. Okay, so most of the repressor are family of what? Heterogeneous nuclear ribonucleoprotein family. HNRNPA1. This is an example of a repressor. HNRNPA1. This repressor bind to the silencer regions. So this HNRNPA1, this repressor bind to a certain silencer region. Here it binds to the exonic splicing silencer element, which is lying within the exon of HIV TAT pre-mRNA and thus represses the inclusion of that exon in the final mRNA. So an example is given here about an repressor protein called HNRNPA1. This HNRNPA1, what it does, this repressor bind to a silencer region. Okay, very good. It binds to a silencer region, and it prevent. Okay, and by binding to the silencer region, it that lies within the silencer region is lies within the HIV TAT pre mRNA within the exon, and it represses the inclusion of that exon in the final mRNA. Okay. So about the HNRNPA1, if I say more specifically, if I say more specifically of this repressor protein, then it would be as follows. This HNRNP1 or HNRNPA1 protein directly actually they bind to the polypyrimidine tract, which is lying located within the intron, and blocks the basic splicing machinery. Okay, like the branch point binding protein BBP, then U2AF, U2 auxiliary factor. It prevents the binding at that polypyrimidine tract, and thus HNRNP1 is also called as polypyrimidine tract binding protein. Okay, so HNRNP1 simply it binds to the polypyrimidine tract and blocks the splicing machinery which initiates the splicing process, like the branch point binding protein, then your U2AF65, that is, which is called U2 auxiliary factor 65. It prevents this HNRNP1. HNRNP1 works in two ways actually to repress the splicing process. It can remove a particular exon from the mature mRNA by binding to the sequence that closely flank the exons and leads to a formation of a loop-like structure as a result of interaction between two HNRNP1 protein found at the end of each exon. So HNRNP1 works in two different ways if you see. Okay, so it can work with two different ways. This is the first way. I will give you a good diagram in my notes. You will see them. The first way is it can remove a particular exon from the mature mRNA by binding to the sequence that flank the exon. This is the exon, not this one. Here HNRMP1 shows the removal of intron. If you imagine the exons here, okay, if you imagine the exons here, that flank the regions. Flank means the sequence which are around the exons. Okay, so it is removing the particular exon. The first way that HNRNP1 works is by removing a particular exon. Wait for a minute. Okay, this one. These are the notes. So wait for a minute. Okay, I have not given the notes here. So first is, yeah, here it is saying. So it can remove or exclude a particular exon from the mature mRNA by binding to the sequence that is around the exon and leads to a formation of a loop-like structure. As a result of interaction, between the two HNRNP1 protein found at the end of each exon. That means what? So the first way is to remove the particular exon from the mature mRNA by binding to the sequence that is around the exon, that is flanking the exon. Okay, very good. And leads to a formation of a loop like structure like this. Leads to a formation of loop like structure like this to as a result of interaction between the two HNRNP. So here we can see two HNRNP is interacting. Two HNRNP is interacting, which leads to a formation of loops that exclude out from the exon. Here I have not said here the intron is removing out, but in this way the exon can also be removed out. Suppose this is the exon and these are the introns. I am marking the introns as pink and the exons as this one. Suppose two HNRNP are interacting, which leads to a formation of a loop like structure like this. Okay, I have to use red now. Okay, it leads to a formation of loop like structure. Okay, so this was your introns, this was your introns, and the middle there is the exon, and the H and RNP1 are interacting with each other, which can cause removal of this exon. Here we have seen the intron is getting removing out by the interaction between H and RNP1. That and this HNRMP1 is interacting where? It is interacting with the splicing sites. Mainly it is interacting with the silencer region, the intronic splicing silencer. Here it is interacting with the diagram that I have drawn just now. It is interacting with the exonic splicing silencer maybe. Okay, so in this way it can remove a particular exon. Just here it is showing that it is removing a particular intron by interaction between the HNRMP1 at specific sites. Okay. In other process, your HNRNP1 binds cooperatively with the other HNRNP1 protein at the mRNA, thereby coating the whole mRNA with HNRNP1 protein across the whole exon. This can render the exon invisible to the splicing machinery. That means, in simple words, you can see that here it shows the intron actually, but I have to show the exons. Okay, let us imagine in case of introns here. Okay, the function, you can able to see, observe the function. The diagram is just opposite of what it has been written. Suppose it's the intron where the HNRNP1 is one by one in a cooperative manner. It binds. It binds to the sites called intronic splicing silencer. When it coats the whole intron, it so HNRNP1 is interacting with one another, and thereby it blocking the 5' splice sites and the 3' splice sites. So the 5' splice site and the 3' splice site is blocked, thereby that splicing process is also blocked. So the whole intron is retained within the mRNA, is included in the mRNA, mature mRNA. So these are the two ways through which the regulatory protein called HNRNP1, which is heterogeneous nuclear ribonucleoprotein, which is functioning as a repressor, is repressing the whole process of splicing. Okay, in this way, HNRNP1 protein works. Okay, so in simple words, if you say, in short, I have written all SR rich protein are activators, and these activators bind to the enhancer region like exonic splicing enhancer, then intronic splicing enhancer, to allow the splicing process to occur. And most of the HNRNP proteins are repressor. HNRNP means the heterogeneous nuclear ribonucleoprotein are the repressors which bind to the silencer region like exonic splicing silencer, intronic splicing silencer, which will repress or stops the splicing process. Okay, just like we have seen about the activators and repressors in previous classes. I hope you have understood that how these protein work together to regulate the alternative splicing. Okay, so this was about all about the splicing process. I hope it is beneficial for you for your syllabus, to complete the syllabus. Okay, if there is, if just go through the whole lecture, I am repeating. You have to study the whole lecture to understand this whole concept of alternative splicing, because many complications has been mentioned here. With the help of example they have shown, like for activators I have discussed about the serine arginine rich proteins. For the repressors I have discussed about the HNRNP, heterogeneous nuclear ribonucleoproteins, I have mentioned. Okay, and diagram is a little bit different here. Instead of showing the removal of the exons, it is showing the intron removal. Okay, so you can write this diagram, don't worry. Okay, so that's for all for this today's lecture. In the next lecture, I will try to discuss the different models of alternative splicing. The different models of alternative splicing that is mentioned right here, I will discuss one of them each with the help of small examples, and then we will finish out the whole alternative splicing process.